So we're Science Policy Exchange. We're a group of graduate students and postdocs from McGill and other universities in the area. We run public events like this and also closed-door events with students and experts to really bring the student voice into policy at the federal and provincial levels. So if there's any students in the room who are interested in joining Science Policy Exchange or getting to know more about us, please contact us at contact@speexchange.ca. If you're on Twitter or on Facebook during this event, pay attention, but if you want to tweet about us, we have our hashtag MyDNAForum for this event. If you want to tweet at us at DSP-SPE. We also have a YouTube channel, Science Policy Exchange, where we have the videos of our last event on personal genetic testing. If you want to look at this, it was a very interesting talk as well. So for this event, I'll let Natalie Brender, our moderator, go through most of the details. But essentially, at the end of the talks, you have a little question card that should have been given to you at the front with a little pencil. So if you can fill that in with a question either for all speakers or for a specific speaker, there'll be SPE members wearing this little badge at the doors on the way out to the coffee break. If you can give them the questions, we'll collect them, sort through them at the front to make sure there's no repeats, and then the moderator will ask the questions to the speakers in the front for the question period after the coffee break. So Natalie Brender was the National Director for Genomics and Society at Genome Canada from 2015 to 2017. Prior to that, she was public policy writer and researcher working with government professionals and nonprofit clients. She was also a research associate and a publications coordinator at the University of Ottawa's Centre for International Policy Studies and a columnist for the Toronto Star on Canada and international affairs. She's also been a policy advisor and speechwriter for Canada's Minister of Foreign Affairs. So I'll invite Natalie to come and give an opening talk.
Thank you, Shawn. Hi, everybody. I am so pleased to be here and extend huge kudos to you and the other organizers of Science Policy Exchange for taking leadership as younger people in this field in bringing policy discussions so intimately into the heart of what is going on in science these days. So there will be a great deal of ethics and legal and science expertise coming up in our panel today. So what I thought I would do is just give a few moments of introduction into the question of what is this general topic that we're all here to talk about and why is it important, and to frame a few broad kinds of issues that you might want to think about as you listen to our two expert speakers. So as you probably know, genome editing is a type of genetic engineering in which DNA is inserted or deleted or replaced in the genome of a living organism. With a kind of molecular scissors. The commonly used metaphor is think of your word processor, where you can take text and you can insert new text or take out the old text or do find and replace. You can do all these things now with gene editing. Versions of gene editing have been around during the past decade, but the technology took a huge leap forward a few years ago with the arrival of the CRISPR-Cas9 system, which allowed genomes to be edited with unprecedented ease and speed and precision, which really changed the game now for how human beings can alter potentially any form of life on the planet. There are two main kinds of potentials for gene editing. One is somatic gene editing, where you change the genetic code in a particular existing living organism. And the other, and sort of considerably more explosive and intriguing, is called germline gene editing, where you change the genetic sequence, change the genes in an egg or sperm or embryo, such that these genes, the changes could be passed on to future offspring. And it's this possibility that raises questions of changing the course of evolution, as people sometimes describe these potentials. This technology, even that we're here to discuss it today and as it pertains to human beings and medical research, but of course it potentially pertains to all kinds of living things, and indeed CRISPR and other forms of gene editing have already been used with respect to plants. There's lots of new kinds of crops being developed using gene editing, which is seen by many people as particularly important in an era of climate change, where we'll need to find ways of coming up with plants that can adapt to climate change. New kinds of animals are being produced, some for food purposes, fast-growing fish, for example, or particularly muscled kinds of food animals for meat. There's also, of course, some novelty uses. Maybe you'll be seeing in the near future genetically engineered little pet pigs or glowing goldfish. You can already get these. And in the broader environmental context, genetic engineering and gene editing is being used to introduce changes into certain kinds of, or potentially be used to introduce changes in certain kinds of insects that carry disease, with the idea that if you can change a mosquito genetic code such that that mosquito that carries disease is also going to be sterile and will not reproduce, the hope is that you can then eliminate diseases that those insects will carry. And so there's hopes of eliminating diseases like malaria or Zika with this kind of technology, at the same time as tremendous worries about other things that could happen when you start doing this. In human beings, researchers are holding out the hope of working on cures for diseases such as Huntington's and sickle cell anemia, preventing children from being born with these terrible diseases. Also treating people who currently have, existing people who have these diseases. And even more ambitiously down the line, there's the thought that gene editing could be used to create immunotherapies for cancer or even cure people of HIV, are some prospects that are being held out. So these are the hopeful aspects, and we'll hear a lot about those. But at the same time, of course, there's also the potential for, on the one hand, removing what's seen to be negative in a living being. There's also the potential for enhancing what's seen to be positive. So you'll probably seen magazine covers with headlines like "Creating Designer Babies," or some people, I think transhumanists, have this idea that they are going to improve on the human being and give themselves through gene editing, powers of enhanced vision or who knows what. But there are also these strains of discussion going on. And some questions then about how to think about these risks and benefits, because they seem so huge on both sides. One important thing to keep in mind is that gene editing is a very new technology for changing life forms, but it's not as though life forms have only begun changing in the past decade with this technology. Life forms are always mutating and changing with successive generations and through evolution. And of course, human beings have been changing life forms deliberately through breeding of plants and animals. So when we're considering what this technology may hold in store, it's not a matter of considering potential harms against the status quo of doing nothing, against the thought of potential benefits this technology could bring. Rather, it might be more useful to consider potential harms from changing life forms, potential benefits, against the background of constant mutation and change in life forms that already is happening. Every time people have a baby in the traditional way, there's new kinds of genetic variations being introduced. So keeping that context in mind is helpful to the discussion, perhaps. As we'll hear, there are, I'd say, two big sets of questions when it comes to thinking about this new technology of gene editing. One is the question about the end states that we envision arriving at. What kind of society might we end up creating if we use this technology, and do we want that kind of society? The other large set of questions is about process. What kind of research would be involved in developing gene editing technology? What kinds of public discussions would have to be involved in deciding how to develop and how to use it? What kinds of regulations and codes of practice would have to be developed? And so you'll be hearing lots about both of these questions, the end state of society we want to be aiming for, and also the processes by which we'll have to develop and think about these. So we have two terrific speakers first to talk about the science, actually, of this technology, and then to open up some questions as to what kind of society might be involved, what might be produced, what kinds of societal implications there are in the process that might be involved in using this technology or thinking about using it. First of all, we will hear from Dr. Alan Peterson, who's an associate professor at McGill with cross appointments in the departments of human genetics and neurology and neurosurgery and oncology. His research program focuses on the developmental and disease mechanisms in the nervous system using mice genetically engineered to model human disease. His lab has provided national platforms for the derivation of such mice and for training in traditional gene engineering technology, and his current investigations are based now on gene-edited mice using CRISPR technology. Secondly, we'll hear from Erica Kleiderman, who is a lawyer and an academic associate at the Center of Genomics and Policy based here at McGill. Her interests center on the ethical, legal, and social implications of access to data and genetic information, regulation of stem cells and regenerative technologies, biobanking, and pediatrics. She's been involved in the coordination and discussions surrounding ethical and policy aspects of the application of CRISPR, gene editing, in both the research and clinical settings. And she's also been involved in efforts to propose amendments to Canada's Assisted Human Reproduction Act. So, enjoyed the discussion, and Alan.